Hello students, now we will start with new topic, that is regulation of gene expression. It means how the expressing genes are getting regulated. In case of eukaryotes, it is of one type, and in case of prokaryotes, it is of another type of regulation of genes. So it means how the expression of the genes are regulated. For suppose, if you take in eukaryotes, during the transcription process, the genes that are responsible for transcription has to be regulated or controlled to some extent. If not, continuously the transcription process keeps on going. So we will divide this board into two parts. In eukaryotes, the regulation of genes happens during first one, transcription at hnRNA level. Because when certain length of one hnRNA is formed, then transcription has to be controlled. Next, and also the split genes number also has to be maintained in constant number according to the species. We know that introns are one of the structures that are nothing but non-coding sequences. So those has to be produced less in number. So in case of split genes, we see one intron is present between two exons. Second one, at splicing step also, regulation of genes will happen. At splicing level of introns, the regulation of genes happens. Next, third one, during the transfer of transcribed mRNA from the nucleus into the cytoplasm of the cell, then also certain number of mRNAs in particular time has to get transferred from nucleus to cytoplasm for the process of translation. So, during regulation of gene expression occurs during mRNA transfer from nucleus to cytoplasm. Next, during translation, the expression of genes is regulated. Expression of genes in mRNA is regulated. Next, in case of prokaryotes, for the gene expression, there is a need of special type of proteins called as accessory proteins. These proteins are coded by special type of genes called as accessory genes. The gene expression is controlled or regulated by accessory proteins. These are coded by special types of genes called as accessory genes. In case of prokaryotes, we see the existence of polycystron, means a continuous DNA double strand will be there, but the DNA fragments which are all binded with each other, those will independently prepare different types of proteins. Means one single complete double-stranded naked DNA synthesizes many types and many number of mRNAs. So we call the type of cystron or the DNA fragment which is synthesizing one type of protein and another DNA fragment synthesizing another type of protein. But all of them are framed into one single-stranded naked DNA in case of prokaryotes. So here the polycystron exists and it is controlled by a combination of genes. What are those genes? Here we said that there are some accessory proteins. So the gene that is responsible to synthesize these accessory proteins is called as accessory gene. So the combination we have written here. So what is those combination? Is a group of or a combination of accessory genes plus promoter genes plus operator genes plus structural genes. All these together are controlling or regulating the expression of genes in case of prokaryotes. A combination of all these is called as operon. Operon term is also given based on the type of expression also, how they are controlling, because when a continuous gene arrangement is there, suppose think that this is A, this is B, this is C, this is D, and this is EG. Now A gene will activate the B gene, the activated B gene will activate the C gene, activated C gene will activate the D gene, the activated D gene will activate the E gene. Now the last one which is activated now again will in turn activate the first gene, that is A gene. So when the genes are controlling or expressing their function like this, one activated by other and the last one is again activating the first type of gene. Such type of gene arrangement is called as operon. Now we will see about operon. As I said you, operon is a group of or a combination of genes. That we will write in the form of definition. So operon is a cluster or a group of genes that code for single mRNA, but that mRNA, it encodes different types of proteins or translates different types of proteins. And many proteins. This condition is called as polycystonic nature. Now, this is a special characteristic feature of prokaryotes. Special characteristic feature of prokaryotes. Now, operon is of two types. Operon is of two types. One is inducible operon. Inducible operon. Second one is suppressible operon. Here, under inducible operon, we take example as lactose. Here, we take example tryptophan. Lactose is a disaccharide that has beta-1,4 glycosidic bonds. But tryptophan is an aromatic amino acid and it is also considered as essential amino acid because it is not synthesized in the animal body. It is an essential amino acid. Lactose is used as an inducer. See, the difference between inducible operon and suppressible operon is normally or naturally the total operon is off in nature, means it is non-functional. But when lactose enters, then it causes the initiation of the function of the operon. Normally, the operon is off, but lactose induces or initiates function of operon and activates it. Coming to suppressible operon. Normally, this type of suppressible operon is functional. It keeps on doing its work. But when the product is more, the obtaining tryptophan is more in the body, then tryptophan itself causes stopping of the operon function. So, normally it is active or functional and is stopped by tryptophan. amino acid. So these are the two types of operons. Now we will see in detail about inducible operon and suppressible operon separately. Now we will see the normal structure of a operon. As I said you, it is nothing but a combination or cluster of genes. So first we can take repressor gene, promoter gene, operator gene, and Z gene, Y gene, and A gene. This is one type of operon. This we can call it as, since here we are seeing Z gene, Y gene, and A gene, we can call this one as inducible operon. Next, another type of operon is there, basing on the tryptophan operon. So here will be the promoter gene. Next is inducible gene. Next is promoter gene. Here also we can mention about existence of one promoter gene. Next, operator gene, same like that of the previous inducible operon. But here in case of tryptophan, there are 5 genes that are regulating or controlling the operon. Here, the structural genes that are responsible for suppressible operon are 5 in number. So 3 genes in inducible operon. Those 3 are structural genes. So we can also write 3 structural genes in inducible operon. 2, 3, 4, 5. In case of tryptophan operon, there are 5 number of structural genes. So all these are called as structural genes. So 5 structural genes in suppressible operon. So this is called as promoter gene of regulator. This is a regulator gene. Means this enhances the function of regulator gene. Now, this is promoter gene to enhance the function of operator gene. So this is promoter of operator gene. This is operator gene. This is Z gene that codes for beta galactosidase. Beta galactosidase enzyme. This is the name of the gene is Z gene. Y gene codes for permease. This is one type of enzyme that allows lactose molecules to enter into the prokaryotic cell. A gene codes for transacetylase. This transacetylase will activate the inactive form of galactosidase enzyme that is synthesized in the inactive form. When this transacetylase is synthesized, it will activate the galactosidase enzyme that breaks the lactose into glucose and galactose. Now here also, this is the promoter gene that induces or enhances the function of regulator gene. Here also the promoter gene that regulates the function of operator gene. So this is promoter of regulator gene. This is repressor gene or regulator gene. In the same way, promoter gene of operator gene, and this is operator gene. In the same way, what are the enzymes that are coded by the structural genes of tryptophan? So E gene codes for anthranilate synthase, B gene codes for anthranilate synthase 2, C gene codes for phosphoribosyl anthranilate isomerase and indole 3 glycerol phosphate synthase. Gene B codes for tryptophan synthase B subunit and A gene codes for tryptophan synthase A subunit. What are these used for? We know that tryptophan is one of the aromatic amino acid. So these types of enzymes are used as flavoring agents in the food processings. Next we will see about inducible operon. We have in detail explanation about inducible operon, that is Lac Operon. Along with that, in NCERT, you have some of the examples. Those are histidine operon, arabinose operon, and Valin Operon. Just the terms are given. Now, under inducible operon, one detailed explanation is given about Lac Operon. But under this, other examples are also there. Those are nothing but histidine operon and Valin Operon. Histidine operon and Valin Operon. Now, another term is also given, that is arabinose operon. Arabinose operon, it functions as both inducible operon and repressible or suppressible operon. Inducible and repressor or suppressible operon. So these are just the examples given. Now we will see in detail about Lac Operon and Tryptophan Operon. Just we will take two examples of those and we will see the explanation. So in case of Lac Operon, E. coli has Lac Operon. E. coli is one of the important bacteria that is residing in the intestine of animal body, or we can say in human body, means in human intestine, and it is important for the absorption of some of the vitamins. In a normal condition, E. coli absorbs glucose from the body. In normal conditions, E. coli absorbs glucose from the food that is available in the intestine. In such conditions, the Lac Operon is off or inactive. But when the intestine is not having sufficient amount of glucose, then if only the lactose is available, still E. coli has to break out that lactose and gain the glucose molecule from the broken lactose and utilize it as its food source. Now, this Lac Operon is having the structures like inducible gene, promoter gene, operator gene, Z gene, and Y gene and A gene. Along with the enzymes that are responsible for breaking down of glucose, E. coli also has an isomerase of lactose. Isomer form of lactose, that is called as allolactose. In normal conditions, what happened? This repressible gene or regulator gene, this is regulator gene, it codes for a regulator protein. Biochemically, this protein is a tetramer. It is having 4 units. In normal condition, when there is no need of lactose, this total protein will go and bind to the operator gene and it will stop the genetic information transfer to the next structural genes, so that the transcription of all this and the translation into the enzymes or proteins will not happen. So it stops operator gene from activating structural genes, so that the translation of the structural genes will not be seen. Here, lactose means the inducer, which is stopping this repressor protein, is absent. So we can write here, this total situation happens when inducer is absent. Now, all the glucose that is available in the intestine is absorbed by the E. coli. Then, there is no more existence of glucose, only lactose is available. Then what is the situation? When complete glucose molecules are utilized, and only lactose is available as a food source, then what happens? The same structural genes I am drawing here: inducer gene, promoter, operator, Z gene, Y gene, and A gene. Inducible gene, it will produce, the regulator gene, it will produce the regulator protein. regulator protein. As we have discussed, it is a tetramer form of structure. Now, when glucose is not available, now the allolactose inside the E. coli body, it will start doing its action because there is no existence of glucose now inside the E. coli body. At that time only, allolactose will enter into the function. So now, allolactose, it will come and bind to the tetramer parts subunits of the repressor protein. They will not allow the repressor protein to come and bind onto the operator gene. So this function is stopped here, so that the operator gene will activate the next continuity of structural genes like this. Now, Z gene, when it is activated by the operator gene, then it produces beta-galactosidase. But this is inactive in form. This activates the Y gene, which produces permease. Now, permease starts at this stage the allowing or the absorption of lactose molecules into the E. coli body. Until the permease is produced, lactose cannot enter into E. coli body, which is residing in the intestine of human being. So, permease now starts absorbing or allowing lactose into the body. Now, this will activate, in turn will activate this A gene to produce trans-acetylase. This trans-acetylase now will activate the beta-galactosidase. So now, this will activate beta-galactosidase. So now, just we will give the numbering for the sequence or the order. So first, this is produced, but here it is inactivated. Next, at the second time, Z gene produces beta-galactosidase. This is the second function. This happens for the second time. Next, the step is production of the permease. Fourth function is permease allowing lactose molecules into the body. Next function is activating. We cannot say next, we have to say simultaneously. It is activating the trans-acetylase, the A gene to produce trans-acetylase. Next, this trans-acetylase, finally it will activate the beta-galactosidase. At the fourth step, lactose has entered into the E. coli body. That will be broken down by the beta-galactosidase. Now, this will break down lactose into glucose plus galactose. Now, what happens? Glucose is now available inside the E. coli body. Now, its body will be in normal metabolic activity. When glucose is sufficiently available, because of more time production of this beta-galactosidase and breaking of the lactose into glucose and galactose again, later, after some time, again the operon can be stopped. So this is about inducible operon. Next we will see about tryptophan operon. It is also called as repressible operon or suppressible operon. Normally, this operon in the E. coli body is active in form because the tryptophan amino acid has to be produced. Tryptophan is an essential amino acid, also one of the aromatic amino acid. Essential amino acid. This tryptophan is also, it is used in synthesis or preparation of melatonin pigment and serotonin pigment. This is also used in synthesis of vitamin B3. It helps in formation or synthesis of melatonin pigment and serotonin. Serotonin, it carries information or it transfers the impulse from the nerves of the brain to the body parts. So this is responsible for synthesis of melanin pigment and also regulates sleep and awake process. Means if a person is suffering from sleep disorders regarding the sleep, this melatonin will be regulating that. Means it controls that disorder. Coming to serotonin, it passes impulse from nerves of brain to body parts. And also, the serotonin also is responsible for the mood, digestion, and also bone health. Also responsible for bone health and sleep and digestion. This serotonin is different from that of secretin. Melatonin and serotonin both are secreted in the pineal gland. This serotonin is considered as precursor for synthesis of melatonin. It is a precursor for synthesis of melatonin. Melatonin is responsible for both sleep and awake process or cycles. Serotonin is majorly responsible for awake process or awake cycle. Then what is the term that is secretin? Secretin is different from serotonin. Secretin, it is synthesized from the S-shaped cells in the duodenum, which neutralizes the acid that has come from the stomach. It is secreted by S cells of duodenum. Sometimes we used to say it is secreted by the intestinal cells commonly. But in depth, if we go, it is secreted by the S cells of intestine or S cells of duodenum. Now, it neutralizes the acidity from or the acid that is secreted from the stomach, the gastric acidity. Now, it also regulates the pancreatic bicarbonate ions. It also regulates pancreatic bicarbonate ions. And it also involves in osmoregulation. If bicarbonate ions are not produced by the pancreas, then bile acids or bile juices are not having this bicarbonate ions. When bile is not having those bicarbonate ions, then its function will not be proper, because sodium bicarbonates, which are produced by the pancreatic juices, are also involved in neutralizing the gastric acidity, and also they are involved in breakdown or emulsification of the fats. So these are the basic points regarding the tryptophan. Now we will see about operon of tryptophan. Repressor gene, promoter gene, operator gene, E, D, C, B, E. Normally, tryptophan has to be synthesized for whatever the functions that we have discussed previously. So these are the genes for structural genes for tryptophan synthesis. When this tryptophan is produced in more amount or its synthesis is exceeded, then the operon will be stopped. So normally this codes for this undergoes this regulator gene, it will undergo the transcription and translation and produces regulatory proteins. But here, this regulatory protein produced in case of tryptophan operon is acting as an apoenzyme. This regulatory protein is inactive in form. Now, it binds to the operator gene, and when it binds to the operator gene, then the information that has to be passed to the next structural genes, it will be stopped, and so that tryptophan synthesis also will be stopped. This total process happens when sufficient amount of tryptophan is produced or more amount of tryptophan is produced than the required. When tryptophan is produced in more amount, then the operon stops. We have seen before what are the enzymes that are coded by the structural genes E, D, C, B, E, anthranilate type of enzymes and tryptophan synthetase type of enzymes. This is about tryptophan operon.